we were talking about the neuroglia of the nervous system, and what the last cell we need to talk about, I think we started talking about, are the oligodendrocytes. And they create the myelin, or the white matter of the CNS, and they also so the same concept happens in the PNS, but it's actually a different cell. So they create this concept of myelination, and what happens is their ends of their processes, they have these processes, and you can see them here. They look kind of um, all of these people, all of these cells look kind of like cyclopses. But what happens is the end of the cell, the processes, the membrane flattens out and wraps around the axon, and it can wrap around the axon multiple times. So the ends flatten out and wrap around the axon. Now, what can happen is you can have multiple cells work together, and so that entire axon can be covered with this this flattened membrane, which at that time is considered the myelin. And if the axon is covered with this, it's considered myelinated. But what can also happen is you can have breaks between where the cells are, where the oligodendrocytes are wrapping around the myelin, and we call those nodes. And nodes are very, very useful because that can speed along the transmission. So the transmission can actually jump from node to node, because that's the one little spot that is not myelinated, not insulated with these particular cells. So the idea behind them is they create myelination, they create an insulation, speeds transmission, and these nodes that you find are useful because they actually make the transmission that much faster. And then in the peripheral nervous system, you basically have two types. You have a satellite cell, and the satellite cell is the helper cell of the ganglia, surround the cell bodies of ganglia and regulate the environment of the ganglia in terms of oxygen, CO2, nutrients, and monitor neurotransmitters. So those are the satellite cells. Satellite cells are also function in the repair of the nerve if it is injured for whatever reason. And then in the peripheral nervous system, it's Schwann cells, or neurolemma cells, that form the myelin sheath. Pretty much the same way, will expand and wrap around, and tend to wrap around in concentric rings around the ganglia, or groups of nerve cells in the PNS. And as I said, nerves in the PNS can regenerate to some degree. It's pretty hit or miss. If in fact a nerve has been injured, if it has not had oxygen or nutrients for an hour or so, which can sometimes happen if it's been injured, or if there's pressure against it, and then all of that is taken care of, and now the oxygen and the blood flow is getting to the nerve. What happens is the axon will repair from the injured portion on down, so the site of nerve damage on down. And this is by virtue of the satellite cells and the Schwann cells. So the Schwann cells will form basically a barrier around it, or a path for that nerve to actually, almost a template for that nerve to repair itself. And this repair in the PNS is hit or miss. It can happen, it's hit or miss, but in the CNS, the regeneration is very limited. What happens is chemicals released by astrocytes, remember the astrocytes are the blood-brain barrier, can block growth, and astrocytes then become scar tissue that fill in the space. So instead of those nerves regenerating, or any kind of nerve cells regenerating in the CNS, you have scar tissue created by the astrocytes. However, what does happen in the CNS instead of regeneration is new connections between cell bodies that weren't there before. Now, on to a different topic, a different part of the nervous system topic. I think you might think of this as we're stair-stepping down into more the nuts and bolts of how the nervous system works. And I didn't do this circling on this, and I'm sorry I didn't know that this was circled when I pulled this up. I pulled this image up because it had multiple bits of information. But what I'm going to talk about now are topics, defining terms, and then we're going to try to pull it all together to explain to you how does that impulse transverse along the axon. So what is an axon what is an action potential? What creates an action potential? So the membrane, so that's the area around, has certain processes that can happen. It can be just sitting in its normal way. We call that the resting potential. And even with the resting potential, also known as the transmembrane potential, what you have is a lineup of positive and negative charges. So you have positive charges on the outside, negative charges on the inside. And basically peace or resting is taking place because there's a balance between the two. And then the other things that can happen, you can have a graded potential. A graded potential tends to be temporary, tends to be very localized change in the resting potential. So you can think of a graded potential as starting on the far left of this blue band and moving just to, say, the potassium leak channel. So it's temporary, localized, and will be caused by some sort of stimulus. So some sort of starting gun will start this graded potential. And then the next one is a full-on action potential, and that's an electrical impulse. It's produced by a graded potential and will propagate along the entire surface or length of an axon, so from one neuron to the next neuron, so the presynaptic neuron to the postsynaptic neuron. That would be what's known as an action potential. Action potentials many times are sum total of many, many graded potentials along the pathway of this of an axon. And then synaptic activity, which is where you have the release of neurotransmitters by the presynaptic neuron or the presynaptic membrane of that neuron, and that produces a graded potential in the neuron that is at the other side of the synapse, the postsynaptic neuron, and then moving on after that. In terms of putting all of this together, you have this concept of information processing, and that is the response from the postsynaptic neuron. Whether that response from the postsynaptic neuron to the action potential that was moved to it via the neurotransmitters are to say, okay, I want to do something with this, I want to send it on, or whether unfortunately it just ends right there. So several things to keep in mind with regard to the topic that we're talking about now is extracellular fluid and intracellular fluid differ greatly in terms of their ionic composition. Concentration gradients of ions of sodium and potassium particularly differ between extracellular fluid and intracellular fluid. And then remember that cell has a selectively permeable membrane, so some things can get through, some things not, And this varies based on many things, but it also varies based on ions. The concentration gradients of sodium and potassium and electrical gradients. So the concentration gradient, so the chemical gradient, and the electrical gradient will separate the charges of positive and negative ions, and the result is the potential difference. So what could happen? So these forces are keeping these positive and negative charges apart, but something can happen to disrupt that. Keeping these two apart. And by the way, this picture, the reason I put this on, this didn't come from your book, but this does show you how a graded potential can move along an axon segment. So you have something coming to change the transmembrane potential, the positive charges moving in, the sodium moving in, and then potassium moving back out in order to create that balance, that resting membrane for that segment, and then having it moving along the entire axon and creating what's known as a full-on axon potential. The opposing forces, or the types of forces that act on transmembrane or resting membrane potential, one of which is an electrochemical gradient. So that's just putting those two together, putting the chemical gradient and the electrical gradient together and calling it an electrochemical gradient. And so the chemical gradient is the concentration of the ions. The electron gradient is the separation, separate charges of positive and negative. So you saw in the last one that potassium and sodium are both positively charged, but they're not the same chemical. So they could actually balance out the resting membrane potential based on their positive charges. So the positive and negative charge can be balanced out, but the chemical gradient is not balanced. So you have both of these types of forces acting on that resting membrane potential, and both need to be kept in check in order for this membrane to not be doing anything, so that for there not to be any action potentials. But for the most part, we want action potentials. Things happen when action potentials happen. In terms of the axon. A few more things that I want to say a little bit more about electrical currents and resistant. Electrical current is defined as the movement of charges to eliminate potential difference. So you have a movement of charges, and then resistance is the amount that membrane says, oh no, you're not going to. So you do have some resistance to that. And then with regard to the chemical, the same thing. Movement of the chemical, and then the resistance, oh no, you're not going to. And each of these are representing what the gradient is for the difference between potassium and sodium, and what the difference would be inside or outside. So upper left, I'll just talk about that one, and then you can kind of figure the rest of them out. What you have is at a negative 70 millivolts, you have potassium chemically having a strong urge to move out of the cell, move from the intracellular fluid to the extracellular fluid. You have electrically potassium with a very small interest in moving into the cell. So potassium chemically, because it's very concentrated inside the cell, so the intracellular fluid, it's very concentrated, so in order to move from an area of higher concentration to lower, potassium is really looking to go out of this cell. And then with the charge, there's not much desire to do that. And then the third part of it is the electrochemical gradient. The net results is that potassium's moving out of here. And we will pick this up on the next one.